Quiz 4, for those of you, I know some of you have taken it already. If you haven't, you have until midnight to get in your 15-minute quiz. And let me remind you about participating. You get points if you do. And I'd recommend that you keep track of your own participation on each class session. So when I send out my record, you can correct it if I miss you. So and I also want to remind you that you need to give your last name when you speak so that I can record that you did. Okay, I want to do
Professor?
Yes?
I have a question. For the quiz, on one of the questions that it was about the isomers, I meant to put formula, but I put formula. I forgot the R.
I'll catch that. I go through each one by hand.
Okay.
If speed grader makes a mistake, I correct it. It's easy.
No, it was my mistake. I didn't put the R in formula.
But it's clear what you meant.
Okay, thank you.
Okay. All right, I want to do a couple of things. One is go over the worksheet from last time, make sure that you have all the right answers. So let me get that up. Share my screen. And you should be able to see that worksheet, the grid. Everyone can see it. I'll make it bigger. Okay, I'll just go through these quickly. There are 16 questions, each in a little box. A DNA molecule is in the shape of a double helix. Proteins are composed of monomers called amino acids. Most DNA molecules are double-stranded, single-stranded RNA. 4 classes are nucleic acids, proteins, carbohydrates, and lipids. The DNA double helix is held together by hydrogen bonds. A peptide bond is formed by a dehydration reaction. Quite a few of you put a saturation reaction, which isn't correct. Remember, a molecule of water comes out for every peptide bond that is formed. And so the loss of water is a dehydration. 4 nitrogenous bases, adenine, thymine, guanine, and cytosine. All amino acids have 2 features in common, a hydroxyl group and an amino group. Phospholipids are critically important in the formation of membranes. Lipids can act as hormones called steroid hormones. Carbohydrates is used to supply energy to the cell. And then there's this formula. It's a formula for a carbohydrate called, and you can use any name. Glucose would be the most common, but any sugar that has that formula would satisfy. So fructose, for example, would also satisfy this. A nucleotide is made up of 3 molecular components: phosphate, 5-carbon sugar, nitrogenous base. Different isomers of certain sugars have different tastes when eaten. In proteins, amino acids are joined by peptide bonds, and the molecule that forms a bilayer that makes up a membrane is a phospholipid. So I show you that for the record. Anyone have a comment or question about the grid?
I've got one, Professor. This is Bennett.
Yeah,
On the one where it's got the formula for glucose, could just the answer simply be just sugar? Or do you want the certain kind of sugar?
yeah, sugar could be any number of things. And not all sugars satisfy this formula. So it would have to be a sugar, a monomer that satisfies this formula.
So it'd be fructose or like this sweet and low type. I forget what it's called.
Sweet and low, I think, is an artificial sweetener, is it not?
It is.
Okay, so that wouldn't work.
Okay. Gotcha.
Okay, anything else? All right, I'm going to stop sharing this screen.
Yeah, actually, Professor, I just had a quick question, please.
Okay, is this Sierra?
Yes, it is, Sierra.
Okay,
On question 8, I got it wrong. The question is, all amino acids found in life have 2 features in common. And you had made like a commentary note, and you had just wrote amino group and carboxyl group. Does that mean that the amino acids, all amino acids belong in either one of those groups?
that means that those two functional groups are found in every amino acid.
Okay. Okay, got it. Thank you.
Okay. Anything else before we move to the main topic of the day? I will share my screen again. And now you see DNA, RNA, transcription, translation, and protein synthesis. Anybody doesn't see that? Okay. All right, so the case in point is cystic fibrosis. We're going to be talking in detail about that soon in the course. And in particular, this one protein, CFTR protein. And the reason this is a good example is that we're going to follow it all the way from its encoding in DNA through RNA, transcription, translation, and finally to the synthesis of the protein itself, and then its journey from the nucleus to the cytoplasm to the plasma membrane, where it's embedded. It's a transmembrane protein, and its function then, when it's in the protein, is to transport chloride ions out from inside the cell to outside the cell. So that's coming up, but it's a good example of this entire process from DNA to protein. DNA replication, that's what we're going to consider first today. When a cell divides, it's got to copy all of its DNA. And so the question is, how does it do that? Well, I'm going to show you a video that describes it in some detail, but this is a preface for that video. There are a lot of terms used, and I want to give you a heads up on what some of these terms are. On the left, you see a DNA molecule in blue. Then you can see that it splits, it opens up. There's an enzyme called helicase, which breaks the hydrogen bonds and separates it into 2 single strands. And these strands, significantly, are called the leading strand and the lagging strand. Now on the leading strand, on both strands, you'll find these SSBs. Those are single strand binding proteins. And they keep the strands separate, keep them from reannealing together. But once this happens on the leading strand, there's an enzyme called DNA polymerase III, which makes a new copy. And it just goes straight from one end of the molecule all the way to the other. On the lagging strand, it's not so simple. And the reason is that it's going backward. And the video will show you why. And it all, well, I should say that in addition to the SSBs on the lagging strand, you have another enzyme called a primase, which puts an RNA primer. Then there is another enzyme. Well, after the primer is there, the DNA polymerase can work and make a short fragment of DNA. It doesn't go from one end to the other, just makes a short fragment. And that little fragment is called an Okazaki fragment. And so there is primer after primer after primer, fragment after fragment. And at the end, there are little gaps between all of these fragments. And those little gaps are filled in by an enzyme called ligase, DNA ligase. Now, there's something I want to emphasize before going to the video, and that is 3' and 5'. That will be referred to over and over again. And I want to show you what that means, because 3' and 5' are critical signals to enzymes so that they know where they are. 3' and 5'. So here you see on the right a nucleotide. On the left, you see 4 nucleotides joined together. The key to 3' and 5' refers to the sugar, ribose, or deoxyribose. The carbons in the sugar are numbered 1', 2', 3', 4', and 5'. So 3' and 5' refer to these 2 carbons, 3' here, 5' here. So on the 3' carbon, there's an oxygen, and then it's hooked to the next nucleotide. On the 5' carbon, there's a phosphate, which is hooked to the next nucleotide. So you see that here. Phosphate joins them, hydroxyl joins them. But then at some point, you come to the end. And if it's at one end, there's a hydroxyl, and that is called the 3' end, because this hydroxyl is on the 3' carbon of the sugar. And if the molecule ends with a phosphate, it's at the 5' end. So that's what 3' and 5' are referring to. And as I said, these ends are critical because it tells an enzyme where it is and whether it can act or not. So, for example, DNA polymerase always starts at the 3' end. I've lost a slide. Okay, I'm going to stop sharing.
So now we'll go through a detailed step-by-step illustration of DNA replication. And what I've done here is I've listed all of the enzymes and proteins that are involved in this process. All of them serve an important purpose. And so we'll go through step by step in order to see how each of them comes into play. So the first step that happens is something called DNA helicase comes. And what it does, it's a 6-piece protein structure. You probably don't have to remember that. But essentially what it does is it unwinds the double helix. So here we have the 2 strands of DNA, and the helicase comes in and it essentially separates these strands. When it does that, these 2 strands serve as template strands, and the process is semi-conservative. That's an important vocabulary word, and what it means is that when we build new strands, we're going to build a new strand here, and we're going to build a new strand down here as well. The original DNA becomes the template strand for each of these pairs. And it's semi-conservative because each new double-stranded DNA segment contains half of the original DNA and half of the new DNA. So it's not fully conservative because you build new components, but it is semi-conservative because half of the new double-stranded DNA is the original DNA strand. So when helicase comes in, it separates the 2 strands, and this forms what is called a replication fork. The replication fork is obviously fork-shaped like that. And what will happen is the replication will now be allowed to proceed on these strands because we have separated them and thus allowed other enzymes to have access. So helicase, it unwinds the helix essentially. It takes the helix and breaks it into its 2 substituent components. And those 2 components then serve as the template strand for the rest of the replication process. Next come single-stranded binding proteins, or SSBs. SSBs are necessary because remember that there is a strong favorable interaction between the base pairs on one strand and the base pairs on the other strand. Remember, these are complementary to each other. And so there is an incentive for them to get back together and bind with those hydrogen bonds. So in order to prevent that from happening, we have these small tetramers called single-stranded binding proteins, SSBs. And the SSBs essentially stabilize these so that they don't end up annealing, joining back together. And that's very important. They prevent the annealing so that these 2 stay separate. And some of them will show up down here like this. Essentially, they just bind to each template strand, each single-stranded template component. And this is important because it prevents the annealing and thus allows these 2 strands to be separate and allows the replication process to continue to move forward. And so SSBs are there to stabilize the strands and prevent the annealing. As the process continues, as the primase and polymerase come along, they'll simply be displaced as soon as the polymerase shows up. But these are necessary in the earlier parts in order to make sure that these 2 strands don't come back together after they've been separated by the helicase. So we've gotten the first 2 out of the way. The helicase separates the strands, and then the SSBs come in and they maintain them in separate components so that they don't come back together and anneal or re-anneal. And then what happens is that DNA polymerase doesn't just start out of nowhere. What it needs is a primer. And a primer is a short segment of RNA. It's a small segment of RNA that will show up, and it will be small numbers. It could be 5, it could be 15 base pairs long. But essentially, a primer is a short RNA component that allows the polymerase to come and bind. So here we'll draw a primer here as well. So the primase shows up in order to produce these short RNA primer segments. And these primer segments then allow for the polymerase to come and start the replication process. So primase creates these RNA primers. They're required to start the replication. And then the big player, which is DNA polymerase, will come along. Remember that the directionality of replication is read up and write down. And so when it's reading from the template strand, it's going to be reading in the 3 to 5 direction. So this primer here will allow replication to continue in this direction. This one, because this is the 3 end and this is the 5 end on this strand, will allow replication to continue in that direction. So replication always starts at the 3 end and moves toward the 5 end of the template strand. The next step then is that DNA polymerase arrives. And this isn't an exactly faithful depiction of DNA polymerase, but you get the essential idea that it's something that connects to the template strand. And what it will do is it will essentially move along and produce more and more DNA. And so the polymerase will move in this direction and it will add bases that are complementary to our template strand. Every time that it encounters one of these SSBs, the SSB will simply be displaced and move away. So here's our polymerase on this end, and it will come along and it has a little attachment there. And essentially this will come along and it will continue to produce nucleotides that are complementary to the parent or template strand. And again, when it encounters the SSBs, they start to disappear. And this process will continue for a while. And so it will just continue to move. Remember that it reads in the 3 to 5 direction, but the new strand will be produced in the 5 to 3 direction. So this will be the 5' end of this, and the new strand will have a 3' end over here that it will continue building toward. Whereas here, the 5' end will be first, and it will continue to build toward the 3' end of the new strand. Remember that this new strand will be anti-parallel to the template strand. So what is the 3' direction of the new strand? It will be going in the 5' direction of the template strand. And so this will continue to happen, and it will build on more and more bases for quite some time. And then we'll get to a point where we encounter one difficulty. And so we'll just build this up to the end or so like that. What is happening here is that as this moves along, the helicase will continue to unwind the 2 strands. And as it does that, what you'll notice is that it will separate. So now let's just say that our parent strand is getting wider. Notice that this polymerase can continue to build into the strand. It will continue to produce new bases, and because the unwinding is simply going to be opening up further components, this top strand can just have the polymerase continue to move, and it will just be producing more bases. The issue is that this other strand, as the fork continues to open and it continues to separate these 2 components, notice that the polymerase is moving this way, and it will expose all of these base pairs that the polymerase isn't able to encounter. And so what will happen is we'll open it up more, and we'll need to build a new primer, and a new polymerase will need to come in. And so we'll get into this discussion of the leading and lagging strand, but recognize that there's a strand that is leading. And what that means is that one primer is necessary, and the polymerase can continue to build as the replication fork opens. The lagging strand is going to open, but notice the polymerase is building in the opposite direction. So these new ones, as it opens, will not get dealt with by this polymerase. And so this is called the lagging strand. And what it requires is it will require, again, another primase to lay down a primer and then to continue that process. And so next we'll get into a discussion of the leading and lagging strand. And that will then bring us into a discussion of RNase H, which is also known as DNA polymerase 1, and DNA ligase. And these are pretty much only necessary for this lagging strand that cannot continuously build as the replication fork opens. And so we'll discuss that. We'll draw this component. We'll redraw it so that the lagging strand becomes clear. And then the discussion will be complete, and you'll understand the replication process how it's fairly continuous in this way, but it's discontinuous in that direction. So what we've done here is we've now advanced this replication fork. So the helicase has continued to unwind these strands. And so it opens up a new region of these two template strands that can now be replicated. And so on the leading strand, it's clear that because the polymerase is moving in this direction, remember it reads from 3 to 5, it can just continue to move in that direction into the replication fork and lay down more and more nucleotide bases. The issue emerges with the lagging strand. The lagging strand has a polymerase that's moving from the 3' end to the 5' end. But what's being opened up is further toward the 3' end than our primer is. And so what we need to do here is we will need to lay down another primer in the lagging strand. And then from that primer, we'll end up getting a new polymerase that shows up. And this polymerase will build on these bases up until it gets to the point where it meets the previous primer. This is called an Okazaki fragment. And it is something that only occurs with the lagging strand because the replication fork is opening and exposing bases from the template strand that are on the opposite side of the direction that the polymerase is going. And this is a discontinuous process, whereas the leading strand can continuously build more and more nucleotides that are complementary to the template strand as the fork opens. The lagging strand is unable to do that. And so you end up with a lot of Okazaki fragments. And this necessitates two other enzymes that we will encounter. And so the first one that we'll deal with is RNase H, or it could be called DNA polymerase 1. And the job of that is to simply remove the primer as the DNA polymerase 3 approaches this old primer. And so what it will do is the RNase H will then essentially eliminate that primer. And then the polymerase, the DNA polymerase 3, will continue to move forward up until it reaches the existing part of the DNA strand. But DNA polymerase 3 by itself is incapable of joining with an already existing strand of newly made DNA. And so what that requires is when the polymerase reaches here, you now need to find a way to link the sugar phosphate backbones of this already existing DNA with the newly synthesized DNA on this lagging strand. And so you need DNA ligase. Ligase is the same root word as ligature, and it essentially means to fasten or tie things together. And so you bring in a DNA ligase, and what that does is it simply, in an ATP-dependent manner, joins these two components together. And so now you're building a continuous strand based off of your template DNA strand. And this will continue to happen as the helicase keeps unwinding this DNA. What will happen is now we will need to lay down another primer, and then we'll build another Okazaki fragment, and it will continue to happen. So the lagging strand requires the use of RNase H and DNA ligase. And that is a consistent theme with the lagging strand. It's a very discontinuous process. The leading strand will only need RNase H. Remember, that's also DNA polymerase 1. It will only need it once because the polymerase 3 is just continuously laying down bases in the direction of the replication fork. So that's the big distinction between the leading strand and the lagging strand. And notice that the process of DNA replication is semi-conservative because we're building a new strand, but we're conserving the parent or template strand. And so it's semi-conservative. Half of this new double-stranded DNA comes from the original double-stranded DNA. There's also another word that we have to use, and this is semi-discontinuous. What that means is that The synthesis of the leading strand is continuous. It just keeps going. The polymerase can continue to move from the 3 to 5 direction. Remember that it's producing this new strand starting over here with the 5' end and going over to the 3' end here. It can continue to do this. And so it's continuous there. But the other half of it is discontinuous. It has to lay down new primer, produce an Okazaki fragment, get rid of that previous primer, and then join the newly synthesized strand with the previously synthesized one using a DNA ligase. And so that is a discontinuous process. So half of it is discontinuous and half of it is continuous. And so that means that the DNA replication is called semi-discontinuous because half of it is discontinuous and the other half is continuous. And as long as you understand that the lagging strand necessitates laying down more primer, having polymerase build more of these Okazaki fragments, and eventually joining them to the other bases we've produced with a DNA ligase, then you'll be able to understand the semi-discontinuous nature of DNA replication. And remember, this is what happens when cells are dividing in, for example, mitosis. And so DNA replication is something that you use not when you're trying to move from DNA to protein, but instead when you're trying to replicate DNA. And usually that is when you're forming new cells or having cells divide, and thus more DNA will be necessary. But remember the leading and lagging strand, remember the different enzymes that can be involved, and remember the semi-discontinuous nature of it, that one of them is discontinuous and the other can just continue producing new bases all the way as the replication fork opens and exposes more of these template strands.
All right, now we're back to 3' and 5'. I took a screenshot of part of his presentation just to preserve for your record the different enzymes in DNA replication and what they do. And I've circled DNA polymerase 3 because the replication of DNA is really just DNA polymerase 3 making a copy of the template. However, DNA polymerase 3 cannot do it alone. It needs these other factors, enzymes and proteins, in order to function. So the entire process requires all 6 of these factors, whereas it's really just one enzyme that's doing all the replicating. I've included a couple of diagrams of the lagging strand because in contrast to the leading strand, the lagging strand replication is quite complicated. And as the man said, it involves laying down new primers. The leading strand only needs one primer for the whole molecule. The lagging strand needs a primer every short distance. And then so the DNA polymerase recognizes a primer and makes a short piece of DNA called an Okazaki fragment. Then it stops and makes another primer and keeps doing the same thing over and over again until you get this large collection of Okazaki fragments. And then another enzyme, the Okazaki fragments don't actually join each other end to end. They need this ligase enzyme to do that. And that's shown in more detail here, where you have the leading strand, which goes on uninterrupted. The lagging strand is interrupted over and over and over again by new primers, new synthesis, and then DNA ligase. Is everybody clear about this difference? It's a very significant difference between how the two strands are replicated. Leading strand is continuous, the lagging strand is discontinuous. And it's because of this requirement of a DNA polymerase to always go from 3' to 5'. If it could go the opposite direction, then you could be continuous on both strands. But because DNA polymerase can only go one way, it has to actually go backward on one of the strands.
So Professor, in the book, it talks about like the daughter strand and the main strand. Is that the lagging strand is the same as the daughter strand? It's just different terminology in the book?
Yeah,
Okay.
yeah, there's only two strands and you can name them, you can name them whatever you want, but yeah, one will be continuous and the other discontinuous, but the same process occurs regardless of what you call it.
Okay, got it.
Professor, I have a quick question. What do you mean when it is continue? Is that going, what do you mean by going back?
Discontinuous means that it's in contrast to continuous. So first of all, continuous starts at the 3' end and the enzyme keeps going. And as the DNA unwinds, it never stops. It just keeps going and going and going, starting with just one primer at the 3' end, keeps going until it reaches the 5' end. And then it's done with that strand. Now, in contrast, on the lagging strand, the polymerase, remember, it's going from 3' to 5', but much of the 3' end isn't unwound yet. The replication fork is moving from left to right. And so the enzyme that's synthesizing new DNA has to start at the replication fork and move toward the 5' end. And in order to do that, as the DNA unwinds, a new primer will be laid down. And so the DNA polymerase can start with that primer, and it goes for a short distance, and it runs into another primer and has to stop. And it isn't until RNase H comes along and takes the primer off that the two Okazaki fragments can join. But then DNA polymerase cannot join them, doesn't have that function. So there's another enzyme that comes along, DNA ligase, which all it does is connect the sugar phosphate backbones.
I have a question, Professor.
Uh-huh.
So when the helicase is formed and the SSBs are there, so they're basically replaced or gotten rid of once you get your Okazaki fragments?
Yeah, the SSBs, single strand binding proteins, he drew them in here. They're only there temporarily, just long enough to keep the DNA strands apart until DNA polymerase can get in there and start replicating.
So when the SSBs are there, they are RNA, and then after they're copied, then it turns back into DNA?
The SSBs are proteins.
Okay.
So they're little short proteins that just bind to the DNA, and their sole function is to keep it from reannealing. But once the enzyme, well, first of all, primase goes in, and remember, the primer is not DNA. The primer is RNA. And the primer is necessary in order for DNA polymerase to start. Once it's started, the primer is no longer needed, and then this other enzyme, RNase, comes along and takes the primer away. And I like the name for it RNase because what it's doing, its substrate is RNA primer.
So Professor, I have a question. So basically the RNA, it's a copy of the DNA, right?
Well, it's complementary to the DNA. It's not a copy.
It's half of the DNA, which is right.
Right.
I just want to clear that.
Yeah, but the significant thing is, first of all, it's needed there in order for synthesis to occur, but it's RNA bonding to DNA, not DNA. It's RNA, DNA.
Oh, okay. And protein makes that copying, right?
The only protein that's not an enzyme is the SSBs, single strand binding proteins. And they don't have any enzymatic activity. They're just a physical presence to keep the two strands after they're unwound, to keep them from snapping back together.
Yeah, that's clear. Thank you.
Professor, I have a quick question on the SSBs.
The SSBs, yeah.
If the SSBs weren't there and the proteins, or not proteins, the DNA did go back together, is that where we start getting mutations where it didn't read correctly?
Probably not. And I'm guessing here, I don't know for sure. But I don't think that this process is inherently mutagenic. We'll talk about mutations later and how they form. And there's nothing in this process that should lead to mutations.
Okay.
One more question, if I could, Professor.
Uh-huh.
Is there any other thing than helicase that would separate DNA?
Anything other than the helicase? In
Yeah,
other words, are there other helicases? I don't know if there is more than one helicase. It takes one, at least one. There may be others that can do it, but something has to separate the DNA. Remember, what's holding the two strands together is hydrogen bonds, which are weak bonds. And it's a good thing they are, because there are so many of them, and they have to be separated in order for the DNA to be functional.
I guess the question that I'm getting to is deleting DNA, can it be split again with another helicase to where, kind of,
I mean, after it's formed a new DNA molecule,
yes.
yeah, it can be split again and replicated again. So that's what happens in cell
It
division.
happens over and over and over again.
Yeah.
Gotcha.
I had a question, Justin Hansen. So on the leading, does it only use one primase and polymerase, like no matter what, or could it also
Yes.
use multiple?
No,
Only one, no matter.
Yeah, there's no need for it. Oops. There's no need for it to start more than once. If we're back here, so this is the leading strand on top, and a primer starts once at the 3' end, and then the enzyme DNA polymerase starts at that primer and then zips down the template strand all the way to the end, to the 5' end, and then that molecule, it's a new molecule of DNA now, and it's finished. So while it's doing that, which is just very straightforward, the lagging strand is having difficulties.
That one. So how many does that one use? Just like practically infinite or whatever?
Yeah, I
Just-
don't know. All I can say is that DNA strands are very long, and so it must be a huge number of primers and a huge number of DNA polymerase molecules. And I have no idea how many. Anything else? All right, he made a big deal about semi-conservative replication. And I put this in just to give you a visualization of what semi-conservative is as opposed to conservative. So this was a big controversy in molecular biology for a long time, whether DNA replication was conservative or semi-conservative. Conservative would mean that you take a double helix and you replicate it, and the products of replication are the original molecule in dark red, and then the new DNA in pink. So that would be conservative, where you're always conserving the original molecule. In fact, it turns out to be semi-conservative, so that you end up, the products that you get, one strand of the product is the old strand, and the other is newly synthesized. And that's true with both of the new molecules of DNA. One strand is old, one strand is new. And we don't need to worry about dispersive. As far as I know, that's never been a serious consideration. But make sure that you clearly understand the difference between semi-conservative and conservative.
I had a question.
Uh-huh.
So is the conservative one the one that used to be like thought to be true and semi-conservative is now what is known to be true for DNA replication? Or do they both work? Like, is that both true or is just semi-conservative true?
Only semi-conservative is true.
Okay.
Conservative never happens.
Okay.
Okay, so we've spent a lot of time talking about how DNA copies itself, which is critical. When we get to cell division, meiosis and mitosis, you'll see that cell division could not occur if DNA copying didn't occur. So then once the DNA knows how to replicate itself, how does information get from the DNA molecule to the protein? DNA is just a storage molecule. It has no metabolic function. It just sits there and waits for somebody to extract information from it. And that's where transcription and translation come in. Here's where we're going. We started with DNA, we're going to a protein, and this is just to remind you what a protein is. It's a polymer of amino acids joined by peptide bonds, and these bonds are formed by a reaction known as a dehydration synthesis, where a water molecule comes out and the carbon atom from one amino acid is joined to the nitrogen atom of the adjacent one, and that new bond is called the peptide bond. Now, this is what all amino acids have in common. They all have an amino group and a carboxyl group joined to a central carbon, which has an attendant hydrogen atom and a so-called R group, the side chain, and that's what causes the distinction among amino acids. So if you just compare tyrosine and glutamine, they both have the same carboxyl and amino ends, but they differ in their side chains. And if we again look at the whole set of amino acids, all 20 of them, there they are. They're all identical to each other, except each of them has a different side chain. Just 2 points I want to make about this. One I made before, and that is that 2 of the amino acids, methionine and cysteine, have a sulfur, each has a sulfur atom. And that becomes critical a little later today when we talk about folding the protein. Another phenomenon is that certain amino acids have hydrophobic groups, like these methyl groups that I have circled here. These are hydrophobic, and these hydrophobic groups can be attracted to each other. And I'll explain to you later how that is, why that happens. But these are the features of amino acids that allow proteins to fold. And they have to fold in order to be active. Again, peptide bond, dehydration synthesis, a water molecule comes out, and this is the peptide bond. It's the bond, the covalent bond between the C and the N, the carboxyl on one side and amine on the other. Now, how does the information flow? So here we have this blue nucleus and a green cytoplasm. In the nucleus is where the DNA resides. DNA never leaves the nucleus. It's always there and nowhere else. DNA is transcribed into RNA, messenger RNA, and the RNA is not confined to the nucleus. It can escape through these little pores, little holes in the nuclear membrane, get out into the cytoplasm where it will encounter ribosomes and transfer RNA. And that's where protein synthesis occurs. So you have transcription in the nucleus, translation in the cytoplasm. Remember that. How many types of RNA are there? 3 that we'll talk about right now. Talk about another one later. Ribosomal RNA, abbreviated as rRNA. There are 2 types, a large subunit and a small subunit. Transfer RNA, tRNA. There are 20 of those, 1 for each of the amino acids. Each amino acid has its own specific and special tRNA, and you'll see why in a minute. Messenger RNAs, there are thousands, at least 1 for each protein. So there are thousands and thousands of proteins in a cell. Each one of those has its own particular mRNA. And down at the bottom here, you see the 3 illustrated. On the right is a transfer RNA, tRNA, and you can see it has a very peculiar shape, but at one end, it has a 3-letter code. It's called a codon, 3 bases. But I want to emphasize that the other end of a tRNA molecule is an amino acid. So for each of the 20 tRNAs, there is a specific amino acid attached to the end of it. And this is what gets transferred to a growing polypeptide chain when protein synthesis happens. Now, on the left is a messenger RNA showing the sugar phosphate background and the 4 bases, A, T, G, and C, lined up. So this would be an unwound, this would be a messenger RNA with all the bases lined up there. And you can see from the color code that there will be matches, complementary matches between the codon on a tRNA and the array of bases on an mRNA. For example, here is purple, yellow, red. Here's purple, yellow, red. So this tRNA would sit on these three. And the ribosome, then it has two subunits, and in between those subunits is the machinery that actually makes a protein. I'll skip that slide. We're getting close on time. So this is how protein synthesis actually occurs. You start up here in the upper left, and you have all three types of RNA: the message, the tRNA, and the ribosomal subunits. And they all come together with the ribosomal subunits sort of clamping on to the message. And then the three-letter triplets call in a specific tRNA, which bind to the anticodon, and they bring in amino acid 1. And then as the ribosome moves down the message, more tRNAs come in, each with its own specific amino acid attached, so that you end up building a chain. Amino acid 1, 2, 3, 4, 5, and it keeps going on until it comes to a stop. Every message has a stop codon in it so that the ribosome knows that's the end of the protein. Then there's a release factor. The protein is released off to do its job somewhere, and the machinery is recycled. It goes back and starts all over again. So then how can you determine what the amino acid sequence is if you know the sequence of the DNA? Well, this shows you how to do it. And this is important. You need to know how to do this because you'll be asked about it in the future. But we'll start with a DNA strand that has this sequence: TA, CA, G, and so on. Now, if it is replicated, in other words, if this is copied to the complementary strand, then T makes an A makes a T, C makes a G, A makes a T, and so on. This strand is complementary to the original strand. So that's how DNA is copied. Now, when it's transcribed, it makes messenger RNA. And now T makes an A makes a T, but no, remember RNA does not have T's. So A makes a U. C makes a G, A makes a, not T, a U. So remember this, when you're asked to transcribe DNA, transcribe it just like you normally would, except change all the T's to U's. And you can see I've underlined each U here in this transcript to emphasize that point. So this is a messenger RNA. It has no T's. Every place there should be a T or would be a T, there is now a U. Well, then the next step, remember, we're on our way to making a protein or peptide. Next step is to translate. And how do we do that? Well, we go to the messenger RNA and we say, what is the first codon? AUG. And that's where you use a codon usage table. So AUG, say the first base is A, that's down here. The second base is U, that's this box. And the third base is G. AUG is methionine. So there's methionine. Methionine is always the first amino acid in a protein. So you go to the next one. it's UCA. So the first base is U, second base is C in this box, and the third base is A. UCA is serine. All of these codons are for serine. So our second amino acid is serine. You go to the third one, AAU. First base is A, second base is A, third base is U. That's asparagine here. You can keep going until you get down to the last one, UGA. First base is U, second base is G, third base is A, UGA is stop. So that's how you go from DNA to a protein, how you can figure out what the amino acid sequence is if you know the DNA sequence. Make sure you understand this, not only from looking at it, but make sure that you can do it yourself. You will be asked to at some time. All right, that's how we get a protein, a polypeptide that has a primary structure. Primary structure, proteins with primary structure have no activity. They're inert. They have to fold, and they fold by forming bonds within the chain. One type of bonding causes secondary structure, which can be either an alpha helix or a beta pleated sheet. And these are always formed with hydrogen bonds. You can see in every case, a hydrogen is bonded to an oxygen. Then there's a tertiary structure, which takes the secondary and folds it even further. And then there's, for some proteins, a quaternary structure. So as I said, this is the primary structure. It's just a long chain of amino acids. It has no activity. Secondary structure forms through the formation of hydrogen bonds, forming alpha helices and pleated sheets. And then we get to the interesting part, the tertiary structure, because this is the form of a protein that has enzyme activity. So how does how does the tertiary structure form? Well, there are four types of bonds: a hydrogen bond, a hydrophobic interaction, a disulfide bridge, and an ionic bond. So you've seen some of these before. You've seen what a hydrogen bond is. You know how that forms. You know what an ionic bond is. Remember the salt, sodium chloride. You have opposite ions attract each other. It's an ionic bond. It forms within a protein chain. Disulfide bridges are significant in that they are covalent bonds and therefore very strong. And then you have this hydrophobic interaction, which is kind of a mysterious attraction that hydrophobic groups have for each other, but it is a force in cross-linking peptide chains. And I'll tell you a little bit about a hydrophobic interaction. So water molecules and hydrophobic molecules, like a methyl group, don't interact. They don't like each other. And so if a hydrophobic, called a hydrophobe, is in water, it will be surrounded by water molecules. In this case, 8 water molecules. And here's another hydrophobe with 8 molecules. But if these two can get together, they now like each other because they've formed a hydrophobic environment. And now, instead of 16 water molecules between the two of them, there are 10. So what they've done is succeed in getting into an environment they like by excluding some of the water. So together, these two molecules have less contact with water, 10 atoms of water instead of 16. So that is what happens when you have a hydrophobic interaction cross-linking a protein. These methyl groups are hydrophobes, and they attract each other in order to exclude water molecules. And now we have quaternary structure. The thing to remember about quaternary structure, it can be either linear or coiled or globular, but it's formed with individual polypeptide chains. So you can only have quaternary structure if you have multiple polypeptide chains, and they're joined to each other by hydrogen bonds and disulfide bridges. And this is an example of one, hemoglobin, where you have 2 alpha chains in purple, 2 beta chains in orange, and the 4 chains together, they're held together, and only in that way can they carry oxygen in blood. And here is a summary of the 4 kinds of bonds that cause temporary or tertiary or quaternary structure. The primary are just the linear amino acid sequence with covalent peptide bonds. Secondary is alpha or beta, alpha helix or beta pleated sheets. These are held together only with hydrogen bonds. And then the tertiary structure has these 4 types of bonds: hydrogen, hydrophobic attractions, disulfide bridges, and ionic bonds. And finally, the quaternary structure is composed of individual, more than 1 polypeptide chains held together by hydrogen bonds and disulfide bridges. Types of proteins, there are structural. It's interesting, collagen is a structural protein. It's the most abundant protein found in mammals. About 30% of us is collagen because it's found in all of these different tissues: bone, skin, muscles, and so on. Spider silk is 5 times stronger than steel on a weight for weight basis. There are contractile proteins like actin and myosin. That's what makes our muscles move. There are storage proteins. There are protective proteins like antibodies. There are transport proteins. We just talked about hemoglobin. Here are red blood cells containing hemoglobin transport oxygen. There are signal proteins like hormones. Hormones are substances that are made in one place and transported to another where they act. For example, insulin. There are communication proteins. For example, neurotransmitters that function between axons and dendrites. Some proteins are toxins for defense or predation. Diphtheria toxin, for example, Clostridium botulinum, is the source of Botox. And then there are enzymes. Enzymes are what make living things live. And we've just talked, when we talked about DNA and RNA, the enzymes that copy them, like DNA polymerase, RNA polymerase, DNA ligase, helicase, and remember, if it ends in ACE, it's likely an enzyme. Virtually all enzymes and the names end in ACE. And the last slide in this set is just a summary that I put together of all the different types of protein and examples of each. This slide set is in your Canvas container. I will put the recording of this session also in your container, or I'll send it to you as an announcement. And that's it. Any questions or comments? I've taken more time than I should have, but I just remind you that if you haven't taken the quiz yet, you should do it before midnight. Okay, I'm going to stop sharing if there are no comments or questions. And this is Wednesday, so I will see you all on Monday.
Thank you.
Okay,
Thank you. Bye.
Professor.
Take care.
all right, take care. I'm going to end the session now.